A very good day to each and every one. I welcome you all once again to Alnas Academy for part 12 of chapter 1 Reproduction in Lower and Higher Plants, class 12 Biology, revised syllabus 2020 of Maharashtra State Board, which also covers NCERT Unit VI Chapter 2. And I am your facilitator Nusrat Siddiqui, to help you and to guide you, having experience of more than 15 years in the field of teaching. Let's begin our video with yet another inspiring quote by John R. Don't let what you cannot do interfere with what you can do. This means that we all have some or the other limitations in what we can do. Use the talents you have, show your passion in what you can do, and don't worry about what you cannot do. Build on your strength in a constructive way. So our today's topic is development of endosperm. So today we are going to discuss about the development of endosperm. In our previous video, we have discussed about post-fertilization events. First, we know that after double fertilization, the event which will be followed will be development of endosperm, that is events of endosperm, followed by development of embryo. Then maturation of ovules into seeds, ovary into fruits are all collectively termed as post-fertilization events. So in post-fertilization events, we have events of endosperm, embryo development, ovary forming into fruit, formation of seeds. This all will be termed as post-fertilization events. Let us see the changes that take place in post-fertilization events. So the parts before fertilization, such as sepal, petal, stamen, style and stigma, they usually wither off and fall off. Then ovary gets converted into fruit, ovule gets converted into seed, egg forms zygote, funicle, which is the stalk of the seed, micropyle, that is ovule. In the ovule, micropyle is present, and that micropyle of the seed facilitate in oxygen and water supply. We know that around the embryo sac there was nucellus cells which get completely used up during the formation of fruit. But if they remain, then they are termed as persistent nucellus, and such seeds are termed as perispermic seeds. Example is beetroot and pepper. The outer integument, it forms testa, which is outer seed coat. The inner integument forms tegment, which is inner seed coat. The polar nucleus forms the endosperm. One of the synergid, it degenerates during double fertilization, and other synergid also degenerates. The antipodal cells which were present at the chelacil, they also degenerate. Let us do a quick recap as what we have studied in our previous video. So male gamete, which is haploid, fuses with egg, which is again haploid in nature, forming a zygote which is diploid, and this process is known as syngamy. Zygote later on develops and forms the embryo. Another male gamete, which is haploid in nature, fuses with polar nuclei, and there are two polar nuclei, they are also haploid in nature, results in the formation of primary endosperm nucleus, that is PEN, which is triploid, and such fusion is called as triple fusion. The primary endosperm nucleus, it divides mitotically to form endosperm. This endosperm is nothing else but it's a nutritive tissue, and it reserves food for the embryo. That's the reason why it is called as nutritive tissue. So in post-fertilization changes within the ovule, the embryo and the endosperm are seen to develop simultaneously. The formation of triploid endosperm nucleus triggered cell division, and that result in the formation of endosperm. On the basis of formation, endosperms are divided into three types. So types of endosperm, the first one is nuclear endosperm, second one is cellular endosperm, and the third one is Helobial endosperm. These are based on the types of formation, how they have been formed, and accordingly they have been divided into three types: nuclear endosperm, cellular endosperm and Helobial endosperm. First we are going to discuss about nuclear endosperm. This is the diagrammatic representation of the embryo sac post double fertilization. That is, the embryo sac contain now the zygote and the primary endosperm nucleus, which eventually gets converted into primary endosperm cell. Now primary endosperm cell undergoes only nuclear division, or karyokinesis takes place. The cell will become bigger, and many triploid nuclei will be formed only through the nuclear division, and this nuclear division is mitotic division. There is no cytokinesis which will take place. So over here you can see the nuclear is dividing, okay, and this nuclear division is through mitotic division. No cytokinesis is taking place, and such endosperm are termed as free nuclear endosperm because there are many free nuclei. Example is coconut water. It is most common type and it is found in 161 endospermic families, and as said earlier, the primary endosperm nucleus, it repeatedly divides mitotically and only nuclear division takes place, and it produces large number of free nuclei. Example is coconut water. Next we are going to study about cellular endosperm. So we already know that post double fertilization the embryo sac is with the zygote and primary endosperm nucleus. So in cellular division, in this type, the division is normal. That means after every karyokinesis, it is followed by cytokinesis. So ultimately a cell mass will be formed which will be triploid, and all the cells thus formed will be triploid cells, and the division is mitotic division. Okay, and in some plant the division of triploid primary endospermic nucleus is immediately followed by wall formation. So the endosperm is cellular right from the beginning. It is mostly observed in the 72 families of the dicot, and example is white edible part of the coconut. Thus in coconut we see that first few division are free nuclear division which results in the formation of water, and then towards the periphery the division is cytokinesis which forms the white edible part. So in coconut we can say there are two types of endosperm, the nuclear endosperm as well as the cellular endosperm. The third type is Helobial endosperm. So we know that in an embryo sac post fertilization we have zygote and primary endosperm nucleus, and it occurs, this Helobial endosperm generally it occurs in monocotyledon. It is intermediate between the cellular and the nuclear type of endosperm. So how this happens? This happens that in this case of endosperm, the few divisions are free nuclear division followed by cytokinesis. Means some cells will undergo free nuclear division and some cell will form cell wall. Okay, so means the first cell division of primary endosperm nucleus will be followed by a transverse wall. Okay, here you can see a transverse wall which divides the cell unequally. The smallest cell will be at the chalazal end and the larger cell will be at the micropilar end. Then the nuclei in each cell will divide by free nuclear division, and then wall develop between the nuclei and the micropilar chamber. So as I said that these Helobial endosperm is intermediate between cellular and nuclear endosperm. The smaller cell will be at the chalazal end and the larger cell will be at the micropilar end. Mostly it is seen in monocotyledon. Apart from the three types, the fourth one is Mosaic endosperm. So three type is the basic type, that is nuclear endosperm, cellular endosperm and Helobial endosperm. Apart from these, the one more type is there which is known as Mosaic endosperm. Now in some cases the tissue of endosperm is not homogeneously distributed, but there are some patches of different colors, and such type of endosperm is called as Mosaic endosperm, and it was observed by Weber in 1990 in Xenia maize. And one typical example which you can see is the example of maize, where the white and red patches appear irregularly distributed. So endosperm containing tissues of two different types is called as Mosaic endosperm, and in plant like corn the endosperm contains patches of two different colors and it forms a sort of mosaic pattern. That's the reason why it is called as Mosaic endosperm. And in case of petunia and tomato, the endosperm shows two types of tissues, some containing diploid cell and some containing the triploid cell, and these two types of cells, when they are intermixed with each other, they form mosaic. So in Mosaic endosperm the patches are not homogeneous, but we see different colors of patches, and in petunia and tomato we see two different types of tissues, some containing diploid cell and some containing triploid cell, forming an mix and these results in the formation of mosaic. Let us have some additional information on endosperm. There is one more type of endosperm which is known as ruminate endosperm. So mature endosperm, when it shows irregularities and unevenness in its surface, then it is called as ruminate endosperm. Now this rumination is caused by the activity of the seed coat or by the endosperm itself, and it is found in 32 families of angiosperm. Example of ruminate endosperm is Alnasacademyindia, palmae and Metaxenia. Next is Xenia. Now Xenia is the type of endosperm where the effects of pollen grain is seen on the endosperm, and it is called as Xenia, and this term was first given by Fouquet in 1881. Example is May. So genotype effect of the pollen is seen on the endosperm. The next one is Metaxenia. So the effect of pollen is seen on the somatic tissue lying outside the endosperm. It is known as Metaxenia, and Metaxenia term was first given by Swingley in 1928, and example is date palm. Endosperm is absent in some of the angiosperm. Example is Orchidaceae, Podostemacea and Trapecia. In some plant, diploid endosperm is noted instead of triploid endosperm. Example is Enothera. Endosperm may persist in the seeds, then it is called as endospermic seed or albuminous seed. Example is castor and cereal. If endosperms are completely absorbed by the growing embryo and the food reserves get stored in the cotyledon, then it is called as non-endospermic seed or exalbuminous seed. Example is pea and bean. A quick recap: we have studied about different types of endosperm: nuclear endosperm, cellular endosperm and Helobial endosperm. In nuclear endosperm, we have studied that only nuclear division takes place, no cytokinesis takes place. In cellular endosperm, after every karyokinesis, it is followed by cytokinesis. The division is followed by cytokinesis also. In Helobial endosperm, it is an intermediate between the cellular and the nuclear type of endosperm, and the division which is taking place in all these steps are all mitotic division. Then we have also studied about the mosaic endosperm, ruminate endosperm, Xenia endosperm and Metaxenia endosperm. That's all for today. Thanks for watching this video. If you have any questions, do write to me at alnasacademyofficial@gmail.com. An entire playlist will be uploaded soon. So till we meet next time, stay safe, stay positive. Do like, subscribe and share this video with your friends so that they may also benefit. Till we meet next time, take care. Bye bye. Thank you and happy learning.